let's see if we can do this. It's about 50 slides, so I gotta go. What? Yeah.
What?
Yeah. Yeah. A lot of them are really stupid slides, like codons are. So we'll see what we can do. Okay. So this is what's going on. Exam on Tuesday. Recitations next week, and it's online. So you do not have to come to recitation, but I will be there if you would like to. So you will not lose 10 points if you don't come, but don't get upset if you have a hard time getting to your paper and you don't ask for help because I was there. Okay. Okay, this is all we've done. This is pretty much like a shorter version of what I gave you guys to work through for the recitation review sessions. What we did yesterday was different mechanisms of action of activators and repressors, and some examples of regulation of gene expression at the levels of initiation, elongation, and termination. And so what we ended with was an example of regulation at termination, which was attenuation. And here what they're doing is, this is only in bacteria because what it's doing is it's using secondary structures in RNA to regulate transcription and whether or not transcription is going to stop early or whether you're going to get transcription through the whole entire operon. And so this is in the trip operon. And this is where, remember, in bacteria, you're going to have transcription and translation happening at the same time. As soon as your RNA is being made, ribosomes are going to hop on and start translating. And so the way this is regulated is by the presence of this leader peptide, and that's just those first few bases, or first few codons in your RNA. They're going to encode tryptophan. And that's a way for the cell to sense whether there's enough tryptophan or not. And so if there's low tryptophan, you're going to get stalling of the ribosome as it searches for the correct tRNA to add. And that allows, in the meantime, you've got the RNA polymerase still doing transcription, right? But that's allowing this secondary structure to form, and that's going to allow transcription all the way through the operon, and you can make more tryptophan. But when you have high tryptophan, there's going to be no stalling because there's plenty of tryptophan available. And so that's going to allow, you're going to actually be kind of obscuring where this hairpin forms, and so that allows this different hairpin to form, which is an attenuator. And so that's going to basically kick off the ribosome before it can transcribe through the whole operon. That's why it's called attenuation. And you won't get transcription of the whole thing. Okay, so what it's doing is it's sensing tryptophan levels and deciding whether or not to carry out full transcription or not. Okay, and this, of course, is regulation of tryptophan operon, which is involved with tryptophan biosynthesis. Yeah.
Is it possible to get the hairpin without that circle bubble thing? Like just have a square rather than circle? Like
Is it possible to get the hairpin without the circle bubble thing? What do you mean? You mean like this circle bubble thing?
Yeah.
So do you think, are you questioning the structure of the hairpin?
Sorry.
I think based on the structure of RNA, it's not a perfect structure, but really what it's kind of showing is that you're going to have base pairing in that stem part, right? And then that looping part is not base pairing, and it's also so close that it can't base pair even if it wanted to, because of the degrees at which RNA can bend. Can it make a square?
Yeah, that's sort of like.
Yeah, I don't know. I mean, square seems like it might be biologically difficult to make, but it's not a perfect loop either.
Okay.
So either way, though, what it's really doing is it's either an attenuator hairpin or not. And so attenuation is going to kick off RNA polymerase, and this other stem loop is going to basically prevent that attenuator from forming, allowing transcription to occur. Okay? Okay. Transcriptional silencing. So how does this happen? Through two major ways. One we've talked about a fair amount is chromatin structure. Can't stand when this door is open. I always feel like I'm on display. Not like I'm not on display, like
Exactly.
but you guys are paying customers now. There's no freebies. No freebies through the door. Chromatin structure, right? We know about silencing through chromatin structure. You know, that's old news. And then it's through these small RNAs. We did a recitation about microRNAs. So you're aware of microRNAs, but this is another form of transcriptional silencing through degradation of mRNA, or through inhibition of translation. So RNAi is a mechanism of targeted degradation of RNAs. And so this is where there's these short double-stranded RNAs that are recognized and cleaved by this protein complex called the dicer complex, and it's cleaved into these very short double-stranded RNAs. And those are loaded onto another protein complex called the RISC complex, where one strand is released and now it's single-stranded RNA. And that single-stranded RNA is going to base pair with the target RNA that's going to be destroyed. So the RISC complex brings the RISC complex plus RNA is brought to the target RNA, and then that target RNA is degraded. And if you degrade your RNA, you're not going to get any protein, right? Everyone agree with me on that one? So I had a question the other day about, well, you say siRNAs, you say microRNAs. What is this about? And I think really the way to think about it is you've got these genes that are going to be encoding these microRNAs that are made, and you can go one of two different routes. But a microRNA is really talking about this kind of stem loop structure of the microRNA, and you're going to get through the activity of dicer and the RISC complex, you're going to get trimming and processing of these small RNAs, and they'll become single-strand RNA, just like what we talked about, and then you're going to get targeting of that RNA for degradation. And then with siRNA, it's a double-stranded RNA that's cleaved. So here you're starting with a hairpin, here it's a longer double-stranded RNA. But in both cases, dicer and RISC complexes are involved in its processing. And in the end, you get base pairing of that small RNA with some target RNA. In one case, you're going to get degradation. In some cases, you're going to get degradation of RNA. And then, I guess I don't show it in a figure, another mechanism of activity is repression of translation through the base pairing with mRNA, right? So you can imagine if you have some kind of double-stranded region suddenly of your RNA, your mRNA, it's going to interfere with translation. Okay, did I see a hand go up? Yeah.
So are you saying that after it passes transcription, like it's just a bunch of transcribed RNA that's getting degraded by siRNA, mRNA? So like is your
Okay, so you'll have like your normal gene expression and transcription, and you get all this RNA. Okay, and that gets made into protein. Right. That's your normal thing that you think about, right? Well, so there's a level of regulation at this post-transcriptional level. And this is where Dr. Ehrlich lost her mind. Okay. She was trying to say level and she couldn't. And she just fell apart. At this post-transcriptional level, you can regulate gene expression by actually degrading your RNA. And so don't think of it as just activate gene expression, tons of RNA, tons of protein. There can also be kind of this fine-tuning of the amount of protein that's going to be made through regulating the stability of the RNA. And that's one of the ways this happens is through the activity of microRNAs and siRNAs. Okay? Yes?
What is preventing a single strand DNA from being translated, like shoved into a ribosome, being translated into a protein? I mean, in terms of coding wise, it's the same except for it has a urine.
What is preventing single-stranded DNA from being shoved into a ribosome? Are you saying DNA? You're asking DNA, right?
DNA, like why? I mean, it's,
What is preventing, this is a lovely question for discussion, what is preventing single-stranded DNA from being shoved into a ribosome and translated? Are you talking eukaryotic, prokaryotic? Because I can answer that different ways. I'm going to let you guys all answer that.
For eukaryotic, ribosomes are in the cytoplasm, so we have to use DNA as a
okay.
Okay, so eukaryotic ribosomes are cytoplasm, DNA is nuclear, so that's going to prevent one thing. Okay. Well, so he's thinking outside the box and asking how come single-strand DNA can't somehow be translated into proteins? Thinking outside the box. So you're saying, well, we need RNA. Well, that's what you're taught.
Doesn't it process like
Unless you have a
What? What are you saying?
I was saying like RNA is processed. and
Okay,
then
so
also
in eukaryotic cells, RNA is processed.
5' cap?
Cap A polydenylation that's required for interaction with the ribosome, for export from the nucleus. But so none of this is addressing bacteria.
But they can't, I mean, because I learned in cancer class, if cells are under stress, they don't need a cap. They can do that without a cap.
Okay, so cells under stress, so no, we need a cap independent translation and export from the nucleus, okay. But you still have introns. You still, okay, so that's the other thing. Okay, so when this is all eukaryotic centric, isn't it? Well, what about, let's make it more simple and go prokaryotic. I just want to see, does anyone on this side, keep on being drawn over here, does anyone on this side of the class want to contribute to this conversation about why single-stranded DNA can't just be bound by ribosomes and translated? Yep.
Well, one thing I can think of is single-stranded binding proteins. Would they be attached and not
Okay.
the ribosome?
Okay, so normally, single-stranded DNA doesn't exist except for when? During replication, right? That's first of all. And when it does exist, it's coated with single-stranded binding proteins. And presence of single-strand DNA for long periods of time does what? Does anyone know? I've said this. Presence of single-stranded DNA for long periods of time in the cell activates a certain pathway. DNA damage response, it's okay. So that's another thing. So like there's so many already, before we even get to it, there's so many reasons that single-strand DNA should never get to the ribosome, but you want to add something?
How about the start codon is AUG and DNA doesn't have uracil?
Okay, so start codon is AUG. DNA does not have uracil. What binds to AUG? Methionine tRNA. And that is base pairing between that tRNA and AUG. Now, you could say that maybe the tRNA can base pair okay with ATG. I don't know. Maybe it's weaker. But it seems like the odds, it's just like the gods are conspiring against translation of single-strand DNA just by the fact that usually DNA does not like to exist single-stranded. My other guess is that single-strand DNA is probably degraded more quickly than double-stranded DNA. But anyone wanted to add to that? But I love these questions because they make you think about things. You can easily go down one path, which is just like, well, it doesn't work that way. But sometimes when you go outside of that and think about it, you actually have to test your reasoning and your thinking skills. So hopefully everyone enjoyed that little exercise. Any other thoughts? Okay. Did I pass your test?
Yep.
Do I get to go get my PhD now? That's like my oral exam. Again, you guys keep doing it to me. Okay. Translation. So, the main parts: initiation, elongation, termination, ribosome, RNA, codons, tRNAs. These are all things that are very important. So remember, we just got our, this again is eukaryotic. We just made our RNA. We've processed it, we've capped it, we've spliced it, we've polydenylated it. It's ready to leave the nucleus, even if under stress, maybe without the cap, it'll leave the nucleus. And that cap, then there's this cap binding complex that binds to the 5' cap and it interacts with the initiation factors that are required for translation, okay? And so we'll get to that. But that's what happens. Out of the nucleus, interacts with initiation factors and ribosomes. So these are just really these are like Bio 201 slides just to get everyone up to speed in case you guys forgot about translation. But hopefully this stuff that's coming up here is you can have a nap while I say it. tRNAs are what link the mRNA sequence with the amino acid sequence, right? Something needs to link your mRNA sequence to your amino acid sequence. And that's a tRNA because it's going to base pair with the codons in the mRNA and bring in the appropriate amino acid. The ribosome is the cellular machinery that translates mRNA to protein. I actually had a question in class in recitation today. It was a conversation. I just want to share it with you. Where is he? He's not here. He usually sits back there. Yeah. Anyways, I'm going to share the conversation. Maybe he'll hear it at home. Where the student was having a hard time keeping straight transcription, translation, and transcription, translation, and replication, and which enzymes are doing what. And he was all jumbled up. And I asked him to ask himself what DNA polymerase does. And so he was just kind of getting confused. And then I asked him to think about what polymerize means. And we talked for a second about how polymerize means to take the individual bases and put them together into a long string of nucleotides. And so DNA polymerizes DNA. So if you know that when you build your long string of DNA, it happens during replication, then you know DNA polymerase is involved in replication of your DNA during S phase. And you're not going to say DNA polymerase does transcription or translation or any of those other things. And so you can kind of go through each of the different enzymes with that same kind of reasoning, and you'll never get it wrong. And so he said to me, wow, that makes a lot of sense. And I was like, yes. And so I want, I'm sure there's at least one person in this room who makes that mistake with transcription, translation. And I want you guys to kind of stop and say to yourself, RNA polymerase. Polymerizes RNA, which means it takes bases of our, nucleotides and puts them together to make RNA. That must be transcription. Anywho, I went on a tangent, but hopefully it helps someone. If it helps one person, then we're good. Okay, so the ribosome, this is what made me think of that story, because the ribosome is the cellular machinery that translates mRNA to protein. So that whole thing started because we were talking about what machinery makes protein. And that's where we got all jumbled up. And so the ribosome is what holds on to the mRNA and brings in your tRNAs and catalyzes those peptide bonds and translates those codons. to make protein. And so you've got your genetic code, that's your triplet code. This is showing your codons, the corresponding amino acids, and the single letter code. People always come into this class not realizing there is a single letter code. I use it all the time. By now you probably realize that, but here's the single letter code. You don't need to know. It's useful if you look at a lot of protein sequence. Notice there's multiple codons for one amino acid. And so that allows for some variation in nucleotide sequence, and it has little effect on protein sequence, right? And that gives the degeneracy that's in the genetic code. Oh, another thing to notice, M-methionine, only one of them, right? AUG, that's your start. And then there's UAA, UAG, UGA, those are your stop codons. Those are good things to know. Translation can occur in different reading frames depending on where your start codon is, right? So for example, this same exact sequence gives you 3 very different proteins depending on the frame that you translate in. And so that's why single nucleotides, like insertion and deletion mutations are so bad, right? Because they change the reading frame. That's also why it's so important to choose the correct start codon, the correct AUG. If you choose the incorrect AUG, you're going to get a completely different protein. Okay, so this is tRNA. And so here's the anatomy of the tRNA. This is the adapter molecule that's going to read the mRNA and bring in the correct amino acid. So it's got the anticodon, and that's reading the mRNA. So that GAA is going to base pair with what? CUU. And up here, here's your acceptor stem, and then at your 3' end of your RNA, you're going to get your amino acid attached there. And then here's the structure, the actual structure. So this is an RNA that's full of these stem loop structures to make this kind of clover leaf looking. Does it look like a clover leaf? A little bit. This kind of clover leaf structure. But in reality, it more looks like this. This is the structure that you're going to see shortly interacting with the tRNA synthase, aminoacyl tRNA synthase. Okay, so I said something about base pairing between the anticodon and codon. Right, so you know what the codon is, that's in your RNA sequence. And then the anticodon is that bottom part of your tRNA that's going to be base pairing and recognizing your codon. And the base pairing is most stringent in those first 2 nucleotides, but the third base is the wobble base, and so that allows for some flexibility in the tRNA codon interaction. And then also in the anticodon, sometimes you'll have an I for inosine, should be I, which forms from deamination of guanine, and it can base pair loosely with multiple bases. I feel like there's a lot of conversation. Does anyone have any questions, thoughts?
Can
Yes.
we go back to the previous slide?
I don't go backwards. No, what?
Oh,
No, it's fine.
I
What?
can just stop up here.
Yeah, no, it's fine.
I was just asking, does this amino acid matches the blue sequence or the green sequence?
Does this amino acid up here, does it match the blue sequence or the green? So what's happening here, and so, this question is going to be very clear once we think about the enzyme that adds it, but essentially, your tRNA is going to bind to an enzyme that is going to recognize your anticodon and going to bring in the appropriate amino acid.
I just didn't know what's the green sequence.
It's just where the amino acid is attached. Yeah, it's not interacting at all with the mRNA. Yeah.
So the different loops, are those all anticodons or just the
No, just the blue is the anticodon. The T loop and the D loop are involved with interaction with structure and with interaction with the ribosome and aminoacyl tRNA synthetase. Yes. Questions? Okay, so you get this flexibility with that third base. That's the wobble that I'm sure you've learned about before. Wobble wobble. And so then also there's this highly modified base that occurs right after, it's a Y here, that occurs right after the anticodon, and that helps with codon-anticodon alignment. Because if you think about it, if this tRNA comes in even at a slight angle, what's to say that, it won't be AAY? It might match up with the complete wrong codon. And so that Y is there to make sure that it kind of comes in and lines up straight down. So there are also different post-transcriptional modifications in the D loop and the T loop that help with folding, structure, and interaction with the ribosome. So this is an L-shaped conformation of the tRNA. So it looks like a clover leaf when you draw it out, but the actual structure is this kind of L-shaped structure. And so it basically spaces, here's your anticodon, the blue, and here's your acceptor stem, and it places it in the right conformation to interact with the aminoacyl tRNA synthetase and with the T ribosomal subunits. So the correct amino acid is attached to the correct tRNA. So this is a big deal, right? The wrong amino acid attached to the wrong tRNA is not good, right? Because then who cares if your DNA sequence is not mutated? The fact that your RNA, your tRNA is bringing in the wrong amino acid is destroying all your proteins, right? So that's a really important part of the whole process. So the enzyme aminoacyl tRNA synthetase is the enzyme that attaches the amino acid to the tRNA. So this, obviously something this important, you would imagine would have some sort of proofreading involved, right? And that's the case. So there's an editing site and an addition site. So you're going to have, here's your tRNAs. Here's your aminoacyl tRNA synthetase. And there's an editing site and an aminoacylation site. So the site where the amino acid is added and the site where it's removed if it's incorrect. And so you can actually have proofreading through a few different ways. One is size exclusion, right? Amino acids, are they all the same size? No. Right, so there's a different aminoacyl tRNA synthetase for each amino acid, each tRNA, right? Because it's specific. It's going to interact, that anticodon is actually going to interact specifically with the enzyme, and the amino acid needs to specifically interact with the enzyme so that the correct match is made, right? So it's not one enzyme just adds like random addition of amino acids. Sorry, I just crack myself up sometimes because then I go into this alternate universe where we're randomly adding amino acids to tRNAs and it gets crazy. Woo! Okay, sorry. It's like spring break up in here. Okay. Oh, you're still stuck on the weather, huh?
Today's wonderful.
It must be that spring break feeling that's making me get crazy over here. Okay. So if the amino acid, so like let's think in our minds about like an alanine and like what's a huge one, like tryptophan. Are they the same size? Who knows all their amino acids? Okay, so only you can. Alanine small. Tryptophan bigger.
It's the lightest one. Is it
Phenylalanine.
the one that has the ring?
Yeah, ring. So what we're doing is we're doing teeny tiny versus huge ring, right? So those are going to not fit into the same space, right? So the first thing is size exclusion. And so the amino acid can't even bind to the enzyme. Okay, so that's one way to make sure the wrong one's not added. And then you can have editing pre-transfer of the amino acid to the tRNA, because if it binds, if it doesn't fit into the amino acylation site, it will be transferred to the editing site and kicked out. Or if you actually do get amino acylation and it's not the right shape and it doesn't fit correctly in the amino acylation site, it will then get pushed over to the editing site and removed. But only the correct amino acid is allowed to be attached and remain attached to the tRNA. This is pretty important if you think about it, because I don't get all spring breaky. So what am I talking about? This was from the old textbook that I stopped using. I like this slide because it kind of shows all the options. It's also fairly busy. So if you're looking at this slide and you're going, I don't know what she's talking about, then what you can do is just go to this slide, which shows it very simply, and then go back to the other one that shows all the options. Right, so you have, your editing site and your amino acylation site, and if an incorrect amino acid gets added, then it'll move to the editing site and get removed. This should be somewhat reminiscent of another enzyme. DNA polymerase, exactly. Totally different. Doesn't really look like an ant. Looks like an avocado. Okay, so there's two mechanisms for ensuring fidelity of amino acid addition, right? One is size exclusion, so the incorrect one won't even fit. And the other one is after addition, it can actually be removed through this editing site. And it all has to do with whether or not the appropriate amino acid is fitting into the amino acylation site or not. Okay. So of course, the correct tRNA needs to be recognized. It's not only about the correct amino acid being recognized, but you also need to make sure the correct tRNA is recognized by the amino acyl tRNA synthetase, right? Because there's one enzyme for each tRNA. And so there's actually a mechanism so that you get the amino acid acceptor arm being recognized up here. So this right here, this green-red thing is your tRNA, and it's actually sitting in the enzyme. And you can see how it's interacting here and over here. And so it's actually interacting with the anticodon, so it's recognizing that anticodon, so that's a level of specificity, right? Because what does the anticodon bind to? The mRNA, right? It binds to the codon, and so that's going to differ in each tRNA. And then it's also recognizing the acceptor arm, that's where your amino acid is added. And so that also is specific for where the amino acid is added. So that's another mechanism for ensuring fidelity. Fidelity, remember what fidelity means, right? The correct thing being added, no straying, no going home with a floozy tRNA. Oh, sorry, we're getting to the end here. You only knew. Okay. Yes.
I'm sorry. Okay, so you said that's another level. So the first level is whether or not the tRNA recognizes the amino acyl.
So the first one is this guy.
Or that's first before.
So the first level, well, if you want to really go back, the first one is whether or not the amino acid even can fit into the enzyme. A second level is the amino acid being added to the correct tRNA? If not, it gets edited and removed, right? Right?
After that, you're saying.
And then here I'm saying that also the correct tRNA needs to interact with the enzyme.
You're saying this happens after that.
Oh God, I don't know about the sequence. It all needs to happen correctly. Don't worry about order, right? Because they're all things that need to happen. The correct enzyme, so there's like 3 specific items involved here, right? Enzyme, tRNA, and amino acid are all very specific to each other, right? And so they all just need to match up correctly so that the correct amino acid is added to the correct tRNA so that the correct tRNA can then add the correct amino acid to the growing polypeptide chain during translation. Okay. Okay, any questions? Good. How are we doing? Okay. So this is just, so amino acids are added to the C-terminal end of the polypeptide chain, right? So you've got your growing polypeptide chain and the tRNA. So here it's showing a tRNA bound to, this is your peptidyl tRNA, so it's attached to the C-terminus of the growing polypeptide chain. And here's your new tRNA coming in. It's called the amino acyl tRNA. It has to do with which bond is there. So this is an amino acyl. This is peptide bond. Is that right? There's a peptide bond here, amino acylated tRNA. And then you're going to get a formation of a peptide bond between your carboxy and your amino group, and that's going to result in the release of the tRNA from the previous tRNA. And so that's how the peptide chain grows. New tRNA, new peptide bond, release of old tRNA. New tRNA, new peptide bond, release of old tRNA. Over and over and over and over again until you hit a stop codon. And then, what's added? A water molecule. It's strange. Okay. So let's talk about ribosomes for a minute. Ribosomes, they're found in the cytoplasm, right? Are they found in the nucleus? Well, not really, not functionally. And they can be free in the cytoplasm or ER associated, right? So when you learn about the rough ER, those have ribosomes attached to them. So why are there free ribosomes and ER associated ribosomes? Well, yeah, but so some are bound to the ER, some are not. But why? What's the purpose of having them either free or ER associated? So it has something to do with protein trafficking and where those proteins that are being translated are destined to go. Who knows where proteins that are being translated into the ER go? Where? Plasma membrane. Where else? Secretory pathway, right? So plasma membrane secreted out of the cell and lysosome, right? And then the free ribosomes in the cytosol are translating proteins that are going to go other places. They'll either be cytosolic proteins, they'll be imported into the nucleus, imported into the mitochondria, imported, where else? Peroxisomes. That's it. Are we satisfied? Have we covered all our locations? Yes. Questions? Were you raising your hand? Yeah.
Yeah, what would happen if ribosomes are in the nucleus? I mean, it will make it much easier having those proteins translate right into the nucleus rather than bringing them out.
So you're wondering about why ribosomes are not just in the nucleus since it seems easier. Why should you have to export things out of the nucleus? Well, I want to see what other people have to say, but I have my own theory. Anyone know why ribosomes aren't just translating proteins in the nucleus? Yeah.
Because is that just where processing occurs and you don't really start translating before you process your RNA?
Oh, so maybe it's some kind of quality control. You want to make sure processing is separated from translation? That sounds like a good idea. Yeah. Compartmentalization allows for higher regulation, higher quality control. What else? Those are kind of things. Yep.
I was just gonna say, maybe like some viruses, if your RNA is in the wrong place, then your body can.
Oh, so detection of invaders such as viruses. If RNA ends up in the wrong place, your body can have an immune response. Actually, that is the case. Certain types of RNAs are recognized as foreign RNAs and destroyed, and they activate the antiviral response. Yep. the ER has a different environment in terms of it being highly reducing, right? Am I right? Yeah, so it has a different chemical environment which is conducive to certain types of protein processing, such as, disulfide bond formation, stuff like that. Yeah.
It might have to do with pH in Europe because when amino acids are connected together, they need to be in certain proteinated and deproteinated state based on the pKa's of each amino acid.
Okay, so when we start thinking biochemistry, in order for amino acids to be attached to each other, they need to be in a certain chemical state, and that's dependent upon the pH. And actually, if you think about it, you're not actually getting translation inside the ER, it's just being translated into the ER. Is the nucleus a different pH than the cytosol? Is it? I don't know. I have no idea, actually. I know there's a pH gradient from cytosol to ER to Golgi to lysosome, but I don't know about nucleus. You'll have interesting questions for me. You're going to be a philosopher. Huh?
I just had the tenderness of blood and wine.
Okay. It's good. It's blood flowing. It's good. Okay, so prokaryotic, eukaryotic ribosomes, they have different components, similar structure and function overall, though. They both small subunit, large subunit, translate proteins. But they're different, which is one of the reasons that antibiotics work, but we won't get any further into that. So the job of the small subunit is to match the tRNAs with codons in the mRNA, and the job of the large subunit is to catalyze the formation of the peptide bonds. So they're both important. Right? Yep. Okay, so if you look, this is the structure of the ribosome, your large subunit and your small subunit, and it's showing the tRNA binding sites. Your A site, your P site, and your E site. A site, aminoacyl tRNA, so that's where your new tRNA that contains your amino acid comes in and binds. Your P site is the one that is holding your growing polypeptide chain, and your E site's your exit site. So as it moves over, your empty tRNA, after it gets detaches from your polypeptide, is going to move into your E site where it can exit. Yes.
So is the large subunit has all those sites on it only, or are the same sites also on the subunit?
Yep. So here's your large and here's your small, and those sites are on both because you're actually having interaction between your large and your small, between your tRNA and your large and small. So they're going to assemble together. So this is, I believe, showing them together. But this is how it looks in a drawing. So you're having binding of your mRNA down here. And then your tRNAs are going to sit there. But they're interacting with your large subunit and your small subunit. Yes?
I don't know if this is for the is there a different ribosome for every type of
Is there a different ribosome for each type of RNA?
like for each type of RNA that wants to be translated, like for the different categories, is it a different ribosome for each category, or can the same?
So ribosomes are fairly conserved. So there's in human genome, there's ribosomes that are made of different subunits and different ribosomal RNAs, and that kind of all come together, and it's one machine. But there's many copies of it. So one group of subunits makes up the ribosome, but there's many, many ribosomes. So if you're asking whether there's some kind of sequence specificity to ribosomes in terms of what they translate, nah. But if you're asking if there are many, 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 many ribosomes translated many RNAs? Yes. Any other questions? Yep.
So this one, this year, is it different here for every amino acid?
There are multiple tRNAs. So if you think about it, I'm going to draw for 2 seconds here. I want you to just think about it for a second. There's a different tRNA for every amino acid, but there's actually multiple tRNAs per amino acid. Yeah, because of the different codons, exactly. Each codon, what are you going to turn on? Here we go. Each codon has its own tRNA. Ooh. That's someone else's thing. Okay. I like that. Whose class was that? The lady with the glasses. Okay. The glasses and the hairstyle. Okay. So if you've got your...
I'm sorry.
There's my clover leaf. Here's my amino acid. And so that's going to be my interaction with my codon, right? Did I? What? Did I draw something dirty?
No.
A little bit. No. Here, I'll do it here. Okay, so there's your clover leaf, right, and your whatever. So there's your anticodon, right? So it's going to recognize a codon. But like, remember, sometimes you've got 4 codons for one amino acid. So that means that your tRNA is going to, there'll be 4 different tRNAs for that codon. And so, yeah, they'll all bind to, for example, serine or whatever. I don't know if that even has 4, but we'll do 3. But they're going to recognize 3 different codons, so there have to be 3 different tRNAs. Make sense? That also means there have to be different amino acyl tRNA synthetases to do that, right? Because they're all going to recognize the anticodon and your amino acid. Yes?
But it's the same ribosome that can translate all 3.
Same ribosome. Same ribosome, because those things are going to come into the ribosome and leave the ribosome. One of these days, the document camera is going to work for me the way I want it to. It always, is a little crazy looking. Yeah.
Okay, so which changing, so that you're saying the ribosome is gonna stay the same, but like the EP and A sites are gonna leave? And those are the ones
The
that are changing?
tRNAs coming in there are going to come here. We're going to go through the whole process. Let's see if we can get there. Yeah.
So the large
You
study.
guys are like, I'm going to make sure there's no translation on this text. You're going to be like, what's the meaning of yeah.
The large
To be happy.
subunit of the ribosome, it catalyzes the.
Peptide bond formation. Yeah. That's true. Yeah. Large subunit, so what did I say? Small subunit matches tRNAs with codons. Large subunit catalyzes formation of peptide bonds. And that makes sense if you look at it, right? What's happening down here, that's your interaction of your mRNA with your anticodon. And what's happening up here, that's where your protein, your polypeptide is. So we'll see. Okay, so this is your mRNA binding site in the ribosome, but we're not going to worry about that. Okay, so let's go to the steps of translation so you guys can think about it. There are movies of this out there, so if you're having a hard time conceptually, you can very easily find movies like animations of this, which is usually pretty helpful. Right, so we've got our E site, our P site, and our A site, and these are each different tRNAs coming in with their amino acid. So in your P site, it's bound to your polypeptide chain. That's your growing polypeptide chain. And so you're going to have your new amino A cell tRNA come into that A site. You always remember what's what because of the letters, right? Exit, polypeptide, amino A cell, right? So that comes in, and then you're going to get your peptide bond forming between amino acids 3 and 4. And so that's going to release it from your number 3 tRNA. And so then your large subunit's going to move over 1 codon space. And then your small subunit's going to move over 1 codon space. And that's going to result in number 3 tRNA being in the E site so it can exit. It can leave, and now there's space for the next amino A cell tRNA to come in. That's the next one. That's going to match with that codon. It's going to be carrying that amino acid, and then the process is going to go over and over and over again until you hit your stop codon. Okay? Yes.
What's happening like pretty fast? It's not like
It's pretty fast.
it's not like
It's not like me talking through it, no.
no, I'm saying it's not.
I don't know what the rate of protein translation is, to tell you the truth. I've done in vitro translation before. You can buy these kits. It's amazing. Where you just put in like your plasmid, and the kits contain all your amino acids and your ribosomes and everything, and you can run this reaction for like 30 minutes and have gobs and gobs of protein. So that's pretty cool. So the peptide bond formation is catalyzed by a peptidyl transferase in the large ribosomal subunit. Okay, so regulation. Question? Oh, you're just like, reach for the sky. Okay. So again, yes.
Is the peptidyl something, that thing, is it combining my 4 and 5 while both are still attached within the A site and the B site?
Yeah, so what's happening is, so like right here, what's going to happen is you're going to get your peptide bond formed here, and formation of that peptide bond is going to release this one. Release the amino acid from the tRNA. I'm sure if we look deeper at the biochemistry, it's probably using the same electrons to do that, which releases your tRNA. Right, and then you have your actual ribosome moving over, which moves everything into the one site over, which opens up a new codon, where a new amino A cell tRNA come in. Okay, so obviously everything's regulated in some way. So here we have elongation factors that are involved in translation efficiency and accuracy. And so in bacteria, they're EF2 and EFG. And then in eukaryotes, it's EF1 and EF2, EF for elongation factor. So in your mind, you should be thinking elongation factor is going to be involved with polypeptide chain elongation. And so what's happening actually is EF2 or EF1 brings the tRNA into the ribosome. And it checks the accuracy of the tRNA amino acid, and it checks the accuracy of codon-anti-codon interaction. And then you get GTP hydrolysis, and then that allows for the elongation factor to release. And so if it's incorrect, it won't actually allow that tRNA to stay. So it's kind of another level of regulation, another level of ensuring fidelity of amino acid addition. Yes?
Is EFTU itself regulated?
However, regulated. Yes, is EFTU itself regulated? I mean, it's regulated in the sense of its GTP or GDP binding, and that's going to control whether or not it can interact with the tRNA. So it brings it in, and if you get a good interaction, strong binding, then it will release the tRNA. So that's, yeah.
Like in the case of cancer, is that EFTU scarce?
So I'm trying to follow your line of reasoning. So you're thinking to yourself, sorry Buzz, you're thinking to yourself that lots of mutations in cancer.
Yeah, and if you have mutations in cancer, you're not really checking that accuracy.
Yeah, but you know what's happening? Those mutations are like at the level of DNA. Yeah, so it's like if your DNA is wrong, then your RNA is wrong, then this could be perfectly accurate, but it's still wrong. Yeah. But I don't know, we should see. Maybe there are more mutations or more dysregulation somewhere in your translation. I don't know in cancer. Okay, so EF2 is escorting the tRNA to the A site. So that little, what is that? A green banana? That it's escorting the tRNA into the A site here. It's GTP bound and it's checking for accuracy. And it's involved with removal of incorrect tRNAs. So if there's incorrect base pairing of the tRNA with the codon, it's going to dissociate. But if it's correct, then you'll get GTP hydrolysis. So now you've got GDP, and then EF2 can leave, and it can leave the tRNA there. Okay. Also, so it's so important that codons and anticodons are matching correctly and that the correct amino acid is brought in. And so you also get this proofreading kind of mechanism done by the 16S ribosomal RNA. And so you actually, the ribosomal RNA interacts with the codon and anticodon. So you've got, and this is involved with EF2 release. So you've got your anticodon binding to your codon, and then that 16S RNA is involved too, and that's stimulating the GTP hydrolysis, allowing for EF2 to leave. Okay, and so if it's incorrect, then you're going to get weak binding between the codon and anticodon and the 16S RNA, and then that tRNA can leave before. Sorry. So if there's incorrect interaction, there's another opportunity for the tRNA to leave. This is now like a third, fourth. I don't know. If we're counting, we've got quite a few mechanisms for ensuring the correct amino acid is added. But so if there's a weak interaction, then the tRNA can leave, even after EF2 leaves. Okay? Yeah?
EF2 is only in bacteria, or is it?
EF2 is in bacteria, but as you can see here, EF1 is the counterpart in eukaryotic cells. So this mechanism exists in both. Okay, so I want you to just kind of think about all the different mechanisms that were used to make sure the right amino acid is added to the growing polypeptide chain, all the way from addition of the correct amino acid to the correct tRNA and the amino acyl tRNA synthetase and all that, to EF2 and bringing in the correct tRNA. There's quite a few mechanisms that we went through. So as you are studying, kind of try to group those and organize those. Okay? Okay. So the ribosome is 2/3 ribosomal RNA and 1/3 protein. So really, it's mostly made of RNA. So the proteins are mostly just used to help assemble and stabilize the RNA structure of the ribosome. So the ribosome is actually a ribozyme, which is an RNA with catalytic activity. Okay, so we talked about elongation. I realized this when I was putting this together, that I teach elongation before I teach initiation, which bothers me because I like things to happen in the order that they happen. But the reason I did that was because the elongation is kind of attached to the theme of correct amino acid addition. So I don't know. Maybe what I'll do is do the initiation long before we even talk about tRNA next year. We'll see. So initiation really requires assembly of the ribosome on the mRNA and scanning to find the first start codon. That's the main thing that needs to happen. Right? So you have an initiator tRNA, which is your methionine, right? And it's going to bind to the P site of the small ribosomal subunit. And it binds to the mRNA by recognizing that 5' cap. Remember, that cap was important, wasn't it? Was. And then it's going to scan for the first AUG. So this requires these EIFs, this is eukaryotic initiation factors. And so the initiation factors are helping with helicase activity. Why do we need helicase activity? Because remember, RNAs like to have secondary structure, and so that ribosome needs to move through the RNA secondary structure. And so you get EIF2, you get EIF4E. Don't worry about memorizing them all. Just understand that there's this order to which things happen, and the goal here is to bring that methionine tRNA to the P site of the small subunit and then start scanning for the first AUG. And that the initiation factor is important for that helicase activity to move through the secondary structure of the RNA. Okay, so once that first AUG is found, the EIFs dissociate, and then the large ribosomal subunit comes in and binds. Now you might ask me, well, how do you know which AUG to use? Because if you look through, like when you're dealing with 4 bases, what are the chances that you're going to come across an AUG, right? Probably a lot. So you might want to have some kind of criteria for determining which AUG to use. And so there are these consensuses. Another consensus sequence, we can have a whole exam on consensus sequences, right? They keep coming up. So there's this consensus recognition sequence that is, did I not say it? That's called the Kozak sequence. That's required for identification of the correct AUG. And this is the sequence right here. So AUG is right in the middle of it. So you could think of it as like what context that AUG is in. And that's where that small ribosomal subunit is going to scan and look for AUG in that context. And if it's not there, then the ribosomal subunit might ignore that first AUG. And this is where I always tell the story of when I was in graduate school and I had to do some cloning, and I cloned this gene, and the gene wouldn't express. I could not get protein. And my boss said to me, well, did you put in a Kozak sequence? And I was like, what's a Kozak sequence? And so now I always tell you, because what happened was I did all this hard work cloning, didn't get any protein because there was no Kozak sequence, so the ribosome just flew right by my AUG. Yes?
Should we know Kozak sequence for the exam?
You should know it for the rest of your life. I mean, don't memorize the sequence, but just understand that that there has to be a way to select the AUG, because there might be 10 of them, and if you choose the wrong one, you're not going to get the right protein because of how translation happens. And so that it's usually in the context of these other, the rest of the sequence, which allows for appropriate AUG selection. Now sometimes you'll have this leaky scanning, which is used to produce proteins purposefully. So sometimes like you'll pass through one AUG in preference for another one, sometimes not, and it'll do this purposefully to produce different proteins with different N terminus. And so, like for example, if there's going to be a signal sequence in one and not in the other, okay? Signal sequences send proteins to certain places. Okay, So this is this is the process, right? So you found your first AUG. This large ribosomal subunit is going to bind and assemble, and then your aminoacyl tRNA is going to bind to your A site. And so then we just kind of go through the process that we talked about before, where you're going to get your peptide bond forming between your methionine and your first amino acid, and then so on and so on. You're going to get movement of your ribosome along, and then this amino acid, the AUG, is going to go into the exit site, and your first amino acid tRNA is going to go into your P site, and it's going to leave an A site open for the next tRNA to come in. Okay, we are almost there, guys. So that all about scanning for the correct AUG. All that was eukaryotic. In bacteria, bacterial mRNAs are not capped, right? Are they? No. Right? And so they need a way to find the first AUG and for the ribosome to bind and enter and start translating. And so they contain what's called a Shine-Dalgarno ribosome binding site, another consensus sequence upstream from the AUG. So this is just showing a whole bunch of genes, and here's your AUGs, and then here's your Shine-Dalgarno sequence. And so if you're cloning, and you're cloning for expression in bacteria, if you put in a Kozak sequence, will it work? No, because that's for expression of proteins, or translation of proteins in eukaryotic cells. If you're going to be cloning for expression in bacteria, you need to have a Shine-Dalgarno sequence. You could think Kozak and Shine-Dalgarno are similar in function, which is they're good for they're required for identification of the first AUG in translation. And Becca, now remember when we were doing the cloning and we talked about this? We were doing cloning in the lab and we were talking about cloning with the flexibility of expressing in bacteria and in eukaryotic cells, and I said, well, let's put in the Shine-Dalgarno sequence and the Kozak sequence, so we got all our bases covered. Anywho, there you go. 2 more minutes. So bacterial ribosomes, they can recognize AUG within the mRNA, right? Because remember, bacterial mRNAs are often polycistronic, where they include multiple proteins, and so you're going to have multiple AUGs, right? And so you're going to have multiple Shine-Dalgarno sequences and multiple ribosome binding sites. Different from eukaryotic cells. So once you hit your stop codon, a release factor is going to bind to the ribosome. So there's no, like if you've ever noticed when you're looking at your codons, there's no amino acid that comes in with a stop codon. It's just stopped, right? And so what actually happens is once you hit that stop codon, a release factor binds, and then you get addition of a water molecule and release of your ribosome. Right? So here's your stop codon, UAG, and your release factor is going to bind to the A site. And then you're going to get addition of your water molecule, and then there's your OH, COOH, and then that's termination. And so here are your stop codons. Your protein is going to be released, your mRNA is released, and your ribosome is going to disassemble. And so release factors are actually an example of molecular mimicry. So the structure of a release factor, even though it's a protein, is similar to tRNA. So it can enter that A site and terminate translation. And so you're going to have polyribosomes, right? Many ribosomes are going to bind and translate at the same time. So you're going to have one RNA, many different proteins made from that one RNA. When you consider the fact that when you express a gene, you can make many RNAs. So you've got many RNAs and then many proteins, you have this kind of like exponential increase in the amount of protein made. And so that's why regulation of protein levels and RNA levels and all that is so important. And that's it, we did it, yay. Okay, so, good luck. If anyone has an extra, multiple choice question, please try to email it to me by, I don't know, like, 4 o'clock. I guess by at least Sunday night, okay?